Yeah. Hi, everyone. Thank you for joining. So I'll begin with this session. Just give me a second. Okay. Is my screen visible to everyone? You can use the chat box or just unmute and say yes, so that I know that you can see my screen. Um, okay, I hope okay, sure. Thank you for that confirmation. Okay, thank you. Hi, everyone. I am Paulami. I am a PhD scholar at IISc and also a PMRF scholar. And this is your problem solving session one for the course Cell Biology, Cellular Organization, Division and Process, which is a course offered by NPTEL. And this is an 8 weeks course, as you all know. And this session, I am sure you would have already gotten the mail. This session is conducted as a problem solving session for you. So if you have any doubts or if you have any questions regarding the course, you can reach out to us. And what we are expected to do is we are expected to do conduct 2 hours session every week. The timing and the slots are fixed. So what we're expected is we have this 2 hour session, and because we this is the first class, so I'll just give you a broad overview of what I generally do in these kinds of classes. But if you would like to do something in particular or some topic that is not clear to you, and you would need some help, do let me know. I will. This chat box is always available, so you can always come and message me here. I can use that part. And what I generally do is that for each week, whatever lecture you have had, so for example, week one, right? So this time you had 4 lectures. So for all those lectures, I give you previous year's assignments practice. You all have exam after the end of the 8 weeks, and you will take an exam, so you need revision. So the idea, the motto of my sessions is that I give you enough question bank so that the topic become clear to you. So I will give you assignment question from the previous year. I'll also give you some assignment question and exam questions from the previous year and some new questions so that you can understand the topic better. And since I have only 2 hours to cover all the lectures of the week, I will just touch upon the topics that have been taught. This is like a more of a problem solving session, doubt clearing session. So I will just touch upon the topics and tell you in brief. The rest of the details and all this are already available to you in the lecture notes. So, but in case if you have any doubt, please do reach out and do message me. Okay. Most of the time I would be in the other screen where I will be presenting from my screen. So I might not be looking in the chat box, but I will keep coming back and checking. So if you have any question, put it in the chat box, or you can feel to unmute yourself and speak to me. Right? Okay, so let me just begin with the session. Right. So as you would have already gone through this, so this course is for 8 weeks, and each week has a particular syllabus that is being targeted. So this week starts with the introduction to cell organization, cellular biochemistry, nucleic acids, information flow, cellular process and division, which I think you would have already seen in the videos that are available. The next week we'll do regulation of the cell cycles. Then the next week we'll move on further into the cell cycle, and we'll go through this ubiquitin proteasome system and S phase, which is basically starting with the cell cycle, following which we'll do DNA replication. Then we'll cover that for three parts. The two of them will be covered in this week, and the third part will be covered in the next week. Then we move on and go to mitosis, which is another part of cell cycle. And then in the following week, you continue with the mitosis, you do cytokinesis, we study about apoptosis, cell death, aging, and then move on to meiosis. And the second last week, we do about meiosis and look at how nuclear organization, chromatin organization takes place, and other such important molecules. And at the last week, we complete the syllabus by looking into all these stages together. So meiosis, mitosis, and then we understand stem cells, plant cells, and budding yeast. Yeah. Okay. And like I said, all these, is my voice not clear? Um, I can anyone else confirm my voice is not coming clearly?
Ma'am, your voice is coming clearly.
Sure, thanks. Okay, I will just maybe move it to a little closer to my screen so that uh, if you have any problem, you can see. Okay, thanks for the confirmation. I hope it's clear now. Else, I think you can just rejoin the chat back. Okay. So you have these eight outlines, and we will be covering each week's course in the respective week. So today is week one, then we'll follow it by week two, week three, and so on. Okay. So in week one, you have these four lectures if you would have gone over the syllabus this week. So you have Introduction to Cell Biology, Cell Components and Organization and Process, which is basically just taking you through what all is part of the term cell. What do you, when you call cell, what all incorporates that term, right? So there was a two lectures in the beginning, and the third and the fourth were about DNA. To first to understand the DNA as a genetic material, what experiments were conducted, and later in the second lecture, we did what are the features of DNA, what makes it the suitable genetic material, right? So we'll follow that theme. Okay. Now, since I told that, uh, sorry, uh, someone wrote then Shushmita, right? I guess it's a network issue on your end, so maybe you can just rejoin. You can disconnect from the call right now and then come back. I hope that clears the sound. Since others are able to hear, I think it's a simple connection issue, so you can just reconnect the call again. Yeah. Sorry, coming back to it. So like I said that I will have more of questions and try to just touch upon those important topics. So let me just begin with the session today. If you have any recommendation or suggestions, please do put. I will incorporate that in the next week. This is all for your help. So I just want the idea here is to give you as many questions as you can practice, right? Okay. So this is how the course also started, right? By describing cell biology and cell. So you would have seen in the lectures also, professor defined first the term cell, right? And then she also explained cell biology. That's what I have also done here. So basically, if you are asked to describe or define cell, you define it as the smallest basic unit of life. And why basic unit? Because it is responsible for all of the life processes. And you will see what processes we are talking about, right? And when you say cell biology, cell is a component, right? Cell is the unit. When you study about those units, this becomes cell biology. So cell biology is therefore defined as the study of cells, which is again the basic unit of life. And we study the structure of the cells. Basically, we look at what are the components of the cell. We look at what are the molecules of the cells, the compartments that are within the cells, and their organization. So cell as a whole. Okay, right? So let's just go on with the cells a bit more. So if you had to give the features of cell, these are the features that you should remember. They are structural, functional, biological units of all living beings. Cells are able to replicate themselves independently. So that's why they are unit of life, because they can replicate themselves, right? Mute. Yeah. Please do unmute. Please unmute when you have to speak, as it's a bit disturbing. Sorry. Yeah. Um, Right. So they can replicate themselves independently, and that's why they're the building blocks, because they don't need anyone else to replicate, right? They can replicate on their own. And when you look at the cell structure, which is right here on your slide, you can see that they contain a fluid, which is called the cytoplasm. They have this membrane, okay? And then they contain several biomolecules in them, like proteins, nucleic acid, and lipids. So these biomolecules, this membrane and the cytoplasm, they constitute the cell, okay? Who discovered these cells? How do you know? How do we know about these cells? It was first discovered, as you would remember in the notes also, in the lecture notes also, professor mentioned the scientist name who are responsible, who are like who are bestowed with the discovery of the cells, right? So we have Robert Hooke, who discovered the cell in way back in 1665, and he observed a piece of bottle cork under microscope, and then he noticed these kind of structures, right? So small rooms. That's how he named it cells, right? And then we also have A.V. Leeuwenhoek, who observed cells and their motility under a compound microscope. And he was the one who saw them as alive entities, right? And then he named them as animalcules. So these are the one, so Robert Hooke saw these structures, and Leeuwenhoek actually saw these structures as a living entity. So we credit the discovery of cells to these two scientists, right? Okay, now, like I said, there were many other scientists that were mentioned. So I've tried to cover those scientists as part of questions. So what I will do is I will stop at these kind of slides which have questions, wait for maybe 2 minutes, and let you answer the questions first, because these are notes which have been discussed in the week, and I'm sure you can do that, and then we can discuss the answers, right? All right. So the first question here is, who discovered cells for the first time? The next question here is, which scientist first observed living cells? These two are easy because we have just discussed them. Then the next question is, all cells arise from pre-existing cells, and this is called also cellulis omnis. And this is a theory which tells how cell replicate, which is what I was covering here. Sorry, this one, right? So these are the term is they can exist from pre-arising cells. Sorry, arise from pre-existing cells. So which scientist gave that term? And we also have cell theory, and we'll see what cell theory is. But you need to tell me first who gave these concepts. You can use a chat box, or you can also try to just answer it for 2 minutes, and then I'll give you the answers anyway. Please do use the chat box if you have answers and if you want to give the answers. Okay, so we have a lot of answers. We have answers for 1, 1, 3, 4, 1, 2, 3. People are answering. We will just wait for 1 more minute for people to answer, and then we can look at the answers together. Right, right. Rudolph. Profile. Okay. Okay, I can see answers, and I can see most of you have, actually all of you have gotten it right. So let me just give you the explanation for what are these, right? Now these two are clear because I just told you in the previous slide. So who saw the cells for the first time? Hook. And who saw them as living entities? Leeuwenhoek, right? Now the third question was, or whichever you take as this question, it says all cells arise from pre-existing cells, and we know this was proposed by Virchow. Now, what does this line mean? It just basically covers the part of them replicating independently, right? And I was telling, this is also called omnicellular as cellular. Now, basically, Virchow, when he saw that these cells can arise from the previous existing cell, he basically rejected the idea of spontaneous generation. So, anytime and every time you study about discovery or anything which has got to biological entities, people, even in evolution, you will see that we have been talking about spontaneous generation, that people just arrive, humans just arrive, animals just arrive on this planet, but and there is no continuity of life. But Virchow, with this theory, told that these cells don't arise independently, or they don't just spontaneously arise. They are continuity of life. They arise from the pre-existing cell, so they continue the information that is there in the previous life. So it's all a continuity of life, right? So he told that. And the last question is cell theory, which was proposed by Sladen and Swan. Now, who were these scientists? What did they do? So, Sladen, also called Matthias Sladen, he was a plant biologist, so he was studying plant tissues, and he found that all the plants are made of cells. Because when he was observing plants, he found these same kind of structures, right? And the other scientist here, Swan, his name is Theodor Swan, he found the same idea in animals. So independently, they found that cells are unit of life for plants and they are also unit of life for animals. So together they came up with the cell theory, which said that all living organisms are composed of one or more cells. One, because they can also be unicellular organisms. Multicellular organisms are us, you, or any other animal that you see around you, right? And these cells, therefore, because they are part of all the living organism, they are the basic structural and functional unit of life. So the term that we used in the first slide, that they are the basic structural and functional unit of life, that comes from this cell theory. So I just wanted to connect the links. Okay. All right. Now, like I said, you have unicellular and multicellular cell. You also have prokaryotic and eukaryotic cells, right? So if you know prokaryotic and eukaryotic cells, you can differentiate them by very visible characteristics, right? So if you see here, this is a prokaryotic cell. If you see here, it's a eukaryotic cell. The first thing that will become very apparent to you is the size. You see eukaryotic cells are very small. So in general, they're around 0.2 to 2 micrometer, and eukaryotic cells are really large. Okay. What is the other feature that truly differentiates them? In a prokaryotic cell, you will never find nuclear membranes. You will find a cell membrane. You will find uh, like important organism or important components, which we will see in the future slides, but you'll not see nuclear membrane being having this very well defined membrane. It will be a nuclear material, whereas in eukaryotic cells, the way we know it, nuclear membrane, and then you have the nucleolus, those kind of structures are present, right? Now, in prokaryotic cells, you will never find, because in prokaryotic cells, you don't find any other membrane other than the overall layers of the cell. So you'll not have these organelles also. So organelles like lysosomes, organelles like Golgi complex, like endoplasmic reticulum, mitochondria, these all organelles which are part of the cells have their own membrane. And these kind of membrane bound organelles are not present in prokaryotes, right? Then what does prokaryote have? What do they have? They have something called flagella, as you can see here, right? They also have glycocalyx, which will be part of their exterior membrane. They have cell wall. That's what I have told. They have their cell wall. They have plasma membrane. They have cytoplasm. Remember, I told you when I was defining cell that they need to have cytoplasm. So they also have cytoplasm. Now, the differentiating component is also about the rhizosome. So the type of, so both eukaryotes and prokaryotes have rhizosomes, large subunit and small subunit. What differentiates is the type of the subunits. You have, when you look at the ribosomal subunits, which are large and small, they have their own density. So it's called at dash S units, right? So 70S, 60S. So those are Swedberg units, right? So that's part of the density. So the density of the two subunits differentiates between the eukaryotes and prokaryotes, right? And as you can see, these are 70S, and we will later see eukaryotes have 80S. Okay, they have a larger size. Okay, and then you have the smaller. So overall, the ribosomes have 70S for prokaryotes. In that, you have the large subunit and the smaller subunit, which have their own Swedberg units. That's also different between these two cells. Okay. Then, like I told you, there is no nuclear membrane. So the nuclear material is just like a globe in the bit in the center, and they don't have histone proteins. Also, they don't have very well defined proteins machinery. And these cells, they divide by binary fission. Okay, and there is no meiosis. Okay, so that's prokaryotic. When you come to eukaryotic, everything that I said is absent in the prokaryotes is present in the eukaryotes. And whatever cells you saw in the first slide, the very well-defined organelles are part of the eukaryotic cells. Okay. You do see cell wall. There is definitely plasma membrane. You see very well-defined cytoskeleton. And then, like I said, you have several histones, because you have several chromosomes. So, and your nuclear membrane is there because of protecting those chromosomes that you have. So basically, eukaryote is a very compartmentalized, highly structured cell. Okay, that's the difference that you can remember for your quick recap. Okay, right. So let's do more questions so that we can understand this much in a better way. So the questions here is this on the slide. So the first one says large subunit of eukaryotic ribosome. Which one is out of these options. Next one is which structure is absent in prokaryotes. This one says which is present only in eukaryote, and this one says ribosomes in prokaryotes are. So be careful with the units. This is also a cue for your exams when if you get questions about ribosomes. First, look at the question. Does it ask about large subunit, small subunit, or ribosomes in general, and then answer about the Sweetberg units. ATS. Right. So TS 60S plasma membrane nucleus. Okay. Plus still typing, so maybe 1 minute more. Anyone else? Okay, people are still typing. Okay. Maybe I'll just give the answers now. Let me see the answers first. Yes. Okay. Okay, I'll just show the answers. Right? Okay, maybe just 1 minute to just tell you the answers and then I'll explain them. So this one says large subunit of eukaryotic ribosome 60S, which structure is absent in prokaryotes nucleus, which structure is present only in eukaryotes mitochondria, and ribosomes in prokaryotes are 70. Now I have seen a lot of ATS in the answers, right? And that's why when I had done when I told you the question, I also told you to look into the units or look into this question specifically. This one says ribosomes in prokaryotes. This one says large subunit of ribosome. So this one is about the large subunit only. This one is all the total ribosome. So uh, be very mindful of when you're answering, right? So just quickly, I will explain to you once again. Like I said, as a Sweetberg unit, it's also the sedimentation or the acidification coefficient. Now, prokaryotic ribosome, the entity ribosome itself, the size of the ribosome is 70S. But when you see the large subunit and the small subunit, large is 50S and small is 30S. And they are not additive. So if the large is 50 and the small is 30, they should together be 80. That's not how it works. So just remember it like this, that size is 70, large is 50 and small is 30. In coming to eukaryotic ribosome, the ribosome overall has size of 80S, the large subunit has 60 and the small subunit has 40S. So remember it like this. Okay. Right. Now, the next question is which is present only in eukaryotes, mitochondria, because mitochondria is a membrane-bound organelle and it is not present in the prokaryote. And here, uh, on a similar line, in prokaryotes, you don't have well-defined nucleus, you don't have the nuclear membrane. So your nucleus is not present in prokaryote, which you all got right. The only confusion that I saw in the answers were about the ATS. So I hope that makes sense, that when I told you to look into the specificities of the question. Now, that's not a problem. It was just a practice for you. So I hope that when you are doing the exams, you are mindful of this. Okay. Now, a very important and a very interesting topic, which is related to the cell walls and the plasma membrane of bacteria. But before I explain that, um, I'll let you have a try at that. So we have two kind of bacteria. We have gram-positive bacteria and gram-negative bacteria. These are prokaryotes. So there are two types of bacteria, gram-positive, gram-negative. What is the basic difference there? So what do you want to think the answer is here? Any idea which one is the correct answer here? Nice. Okay. Uh, answer one. Okay. Okay. Um, right. Okay. So it's a very good like revision exercise for you. So in case you are like whenever you are revising, that's how the idea is that whenever you are revising for your exams or whenever you are revising the course, these questions can help you as a guide. So you can always use them as your tracker. Okay. Okay, so people are already answering, and I guess we have given a lot of different kind of answers. So let me just give you the answers first. Okay, so we have gram-positive bacteria which appear purple. The appearance of purple is due to the retention of crystal violet iodine complex, also called CVI complex. And the next one is gram-negative bacteria, and this have outer membrane with LPS lipopolysaccharide. Right? It's very simple idea. This is the coloration that we are talking about. It's part of an experiment called gram staining. But the gram staining basically explains the difference between gram negative and gram negative. The name gram negative and the gram positive comes from the result of the gram staining. Okay, so let me just first explain to you what does it mean. So this is how a bacterial cell wall looks like. This is G plus means gram positive, and this is G negative means gram negative. Okay, now if you see, so they have plasma membrane, right? And I told you they have a plasma membrane in the cell wall. And I told you they have a plasma membrane in the cell wall. So you have a thick cell wall. Okay. This cell wall layer is made up of peptidoglycan layer. Okay. This is a peptidoglycan layer. And what is a peptidoglycan? It's basically, as the name suggests, it's a peptide and it has also sugar residues. So it is made up of these NAGs and NAM molecules or polymers of these molecules, and they are linked by beta 1 for linkage. Okay. And on this polymer, you have, so basically these are polymers which have sugar attached on a peptide chain. So peptidoglycan. So peptide chain, which is composed of the amino acid, alanine, glutamic acid, lysine. These are all different amino acids that make up a peptide, if you remember. And then on that, you have the sugar molecules, NAM and NAGs. Okay, and these are cross-linked with each other, so they form a very thick layer. Okay, so gram positive, just remember it like this: they are positive and negative in terms of the peptidoglycan. Peptidoglycan thick means they are gram positive. If you see the gram negative bacteria, you have this layer, but a very thin layer. On top of that, you have this lipopolysaccharide layer. Right, that's what is important. Now, why did they get the right? I see someone is writing both right. So I was telling you guys already answering everything so correctly. I'm glad, and I hope this session is just helping you to understand this a bit more. Right. So let me just quickly explain the principle of gram staining, which is a very common experiment that people do in your masters, in your bachelor's, when you come to even further research. If you're working in a microbiology lab or anything that has to go to bacteria, this is one of the first few experiments that people are taught, right? So let me quickly explain the principle behind this, right? So gram staining is basically a technique to classify them, the bacteria as positive or negative, and the you have already got the idea where the coloration would come because there is a difference in the structure. Now, where is the purple color coming from? So the bacteria which have the thick peptidoglycan layer, they, because of the cross-linking between their polymers, they are able to retain the crystal violet iodine complex, so CVI complex, right? And the other bacteria is gram-negative bacteria, because they have a thin peptidoglycan layer, they are not able to retain the coloration. So when they don't retain the primary stain, they retain the counter stain, that is saffronin, and that gives them the red color. Okay, and that's why you observe them in two different coloration. So if you, this is basically a flowchart of the experiment. So first, what you do is, first stain that we add is crystal violet. Okay. Now, anything that is gram negative, anything that is gram positive will retain the CVI complex. Okay. After crystal violet, you next add iodine so that it's crystal violet iodine form a complex and they become this large blob in your peptidoglycan layers. Right. Third step is alcohol, which is a decolorization colorizer. So if in your thick peptidoglycan layer you have the CVI complex as a large blob, these decolorizer can't move them, so they don't lose color. But the gram-negative bacteria, which have very thin layer, and they have lipopolysaccharide. So alcohol will dissolve the lipopolysaccharide and then will wash away the color. Whatever remaining is also washed out, right? And then you give this counter stain saffronin, which colors the gram-negative bacteria as pink color. But gram-positive bacteria, because they're already purple in color, they don't show up as pink. So that's why under the microscope, after this experiment, when you see something as green, something as pink color, they are gram negative, and then something as purple color, they are gram positive. Okay, and what is the importance of this layer? Why are we even talking about this layer? So this layer serves as a very important target for antibiotics. So antibiotics like penicillin, they are basically targeting these kind of layers to kill the bacteria. So these are a target for the drugs that we have, that we use to kill the microbes. Okay, right. This is very interesting, and I think I hope it was clear to you. Now, quickly coming to cell organelles, I said I will not go into too much detail. I will just quickly tell you different kind of organelles that you have in your cell. Of course, you have nucleus, which basically stores genetic material, and they are the site of replication, transcription. This we will continue and cover in great details in the next few lectures. You have mitochondria, and everyone in this world probably calls it as the powerhouse of the cell, rightfully so, because it is a site of ATP production. Uh, then you also have endoplasmic reticulum. You have two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. Uh, rough ER is site of protein synthesis, smooth ER is site of lipid synthesis. Okay. Then you have Golgi apparatus, which is basically involved in protein trafficking. So they modify, sort, and package the proteins and lipids, and they secrete these molecules out of the cells if needed. Okay. Then you have ribosomes, the ribosome that we have just discussed in great detail. These are site of protein synthesis. And the free ribosomes, these are making this cytosolic proteins. You also have bound ribosomes, which are required for membrane proteins or secretory proteins. Okay. Then there are some other organelles like lysosomes, peroxisomes, and these have enzymes and they have their own um, cell processes going on in between them. So for example, lysosomes, they have these intracellular digestion. So anything that has to be killed off in a cell is killed off or is degraded in the lysosomes. And then you have peroxisomes, which are important for fatty acid oxidation, which is basically your detoxification of the harmful substances which enter the cells, and the cell takes all the measure to save yourself. Right. And then if you go to plant cells, will have vacuole, you have chloroplast. So all of these are there. And the next one, the other one that I will say is cytoskeleton, which also something that we mentioned that is lacking in prokaryotes. Cytoskeletons are basically networks of microtubules, microfilament, and intermediate filaments. These terms will again crop up when we are studying about cell cycle, when we are talking about anaphase, telophase, all these different phases. These cytoskeletal components like microtubules, microfilaments will come up there and we'll understand them better there, right? But for now, you just remember that these are the different kind of organelles that are there, right? Now, if you remember your lecture notes, professor also mentioned that you have different kind of molecules in the sense, right? You have macromolecules, you have micromolecules, you have molecules with different kind of compound bonds between them. So those topics, I thought I'll just cover it some questions, and that would be good revisions for you. So these are, I think there are 8 such questions. So I just try to cover and bit of organelles also in this, right? The first one is which of this is a macromolecule? When you talk about nucleic acid, which is again a biomolecule, what are they consisting of? What are the units of? Uh, Lipids differ from other macromolecules in what aspect? And the powerhouse of the cell corresponds to what, which is everyone knows. So you can give an attempt and we can see how much. I'm sure these are very easy questions, so this shouldn't be very difficult. Right. Made of monomers, which one? Okay. Yeah, please do write the answers. I've just gotten 1 or 2 answers. Just wait. Okay, so protein insoluble in water. Yes, MCQ, yes. The answers, the questions in the exams are mostly MCQ type. You might have 1 or 2, which would be like one word, like fill the blanks kind of question, but that will again be MCQ. So mostly it is MCQ. Okay, so most of you have given me answers. So let me just quickly go again. Yes, like everyone mentioned, and I keep telling you guys are getting it all right already. So macromolecules are protein. Lipids differ in terms of other macromolecules because of being insoluble in water. Nucleic acids are composed of nucleotides, and powerhouse of the cell is mitochondria. This answer is, I was pretty sure you'll get right. Just for few people who did not get these rights, I will just quickly go over. So macromolecule question, the answer is protein. That's because protein are composed of different like different units, biomolecules, right? So they are usually polymers. So macromolecules, macro means they are polymers, and proteins are composed of different amino acids. So amino acid is the unit. So any unit is a monomer, right? So because amino acids keep repeating and making a protein, protein is the macromolecule. That's what amino acid is not. Okay. Water is a small inorganic molecule. So that's not a biomolecule, that's a small inorganic molecule. And coming to glucose, glucose itself are again monomers. When glucose are like when they bond with each other, they form different kinds of compounds like sucrose and galactose, right? So those make up. So fructose, for example, galactose can be called a polymer because they have multiple repeating units, but glucose is one single monomer, right? So that's why it's not a macromolecule. And coming to lipid being different from macromolecule in terms of their insolubility of water, that's because lipids are hydrophobic. And that's again because of the hydrocarbon chains, if you remember the structure. And because of these hydrocarbon chains and because of being hydrophobic, as the name suggests, uh, basically phobia means not liking, and hydrophobia means not liking water. So these don't get solubilized in water. They don't dissolve in water. Whereas polymers, they are also polymers. Lipids are fatty acid polymers. So basically, lipids can what do you say? They don't differentiate with others in terms of being a polymer. They are polymer, right? They are not hydrophilic at all. So they are insoluble in water. That's the correct word for lipid. And they are not monomers. I mean, in the sense that they are uh, also, they will make up something else, right? Okay, right. So everyone got here also correct, which is great. Okay, next set. So protein synthesis, which is I think I already covered. Protein synthesis occurs in which organelle? Detoxification of drugs occur in which organelle? Organelle, which is responsible for intracellular digestion, and dash are formed when electrons are transferred from one atom to other. This covers the bonds that the professor was speaking in her notes. And this will be also a good revision for you to remember all the different kind of bonds that she had mentioned in the lectures. Okay, so ribosome, lysosome, what else? Smooth ER. Ionic bond, right? Okay. Anyone else wants to give a try? Ionic bond, okay. SER, smooth ER, lysosomes. Okay, all right. Okay. That's great, like really great. Everyone is already very well aware. Right, so it's correct. Protein synthesis occurs in ribosome. Organelle occurs, organelle, which is responsible for intracellular digestion, is lysosome. Detoxification of drug is in smooth ER. We already discussed this. And the last one is ionic bond. I am sorry, this just got, I don't know what happened here. Just a second. Uh, Anyways, so you got that right. It's ionic bonds, right? And now just I will quickly maybe just tell you just a second. Yeah. Uh, just quickly for revision, what are the other and what does ionic bond actually mean, just in case anyone was confused. So just for your revision, ionic bonds are basically those bonds, as you can have already answered. These are when an electrons are completely transferred from one atom to other. So you need a cation, you need an anion, and they share the bond by electros. The bond is held by electrostatic interaction between these opposite charges. So that's for ionic bonds. Okay. The other bond that I have mentioned, like for example, covalent bond, it's basically there is no transfer of electron. The electrons are shared between these two units. Okay. Hydrogen bond is basically a weak attraction between partial charges, and it has to involve a hydrogen. Okay. And peptide bonds, which is again specific covalent bond, which are specific for amino acid, which is what makes up the chains of your protein. Okay. Right. For your revision. Okay, now another topic which is also very widely discussed in cell biology is the plasma membrane. And I hope yeah, sorry, I need to just a second. I guess someone has unmuted themselves. Uh, okay, all right. Um, Sorry for that interruption. It's just that when there is a this thing, I just have to come back and check. So sorry about that. All right, sorry. Come back to it. coming back to the topics that we were discussing, coming back to plasma membrane, which is again, like I told, is a very widely discussed topic in cell biology. And that's because the structure of plasma membrane was under a huge discussions, and many people have their own suggestion for how the plasma membrane looked. So plasma membrane has its own history. So if you so basically, when you talk about plasma membrane, you define it as a thin, living, selectively permeable boundary, and that's what makes it the most important cell boundary because it is selectively permeable. Everything and anything cannot just enter the cell. It is guarded by its plasma membrane, which only allows certain molecules to pass inside the cell. Okay, and they enclose the cell, and that's why they also regulate the exchanges of substances between the cell and its environment. Now, the cell membrane or the plasma membrane concept itself underwent several structural changes. For example, the first structure was suggested by Overton, who said the cell membranes are completely lipid in nature, right? And the permeability of the plasma membrane will depend on which kind of lipid soluble substance will come. If they are lipid soluble, they will enter the enter. That's Overton's version. Okay, and this is somewhere around 1890s or something like that. Okay, and then came the next model, which was given by Gotter and Grendel, which said where we get the lipid bilayer model. Okay, and they said that yes, there is lipid, but that is lipid bilayer. So that's not just a single layer, as you see on the slide right now. These are basically bilayers. So you have one lipid facing up, one lipid facing down. This one is extracellular outside the cell. This is intracellular towards the cytoplasm. Okay. So Gordon and Grendel said that yes, you have two lipids which are part of the plasma membrane. This was somewhere around 1920, 30, somewhere like that. Similarly, around that time, you also have Daniel Neffson's version, which now called, no, it's not just lipid, it's actually a protein lipid protein layer. So you have a protein, then you have a lipid, and then you have a protein. So it's called the sandwich model. And that it where they said that is you do see lipid bilayer, but you have protein on both sides. So basically what they meant was on this plasma membrane, you have protein at both sides of these lipids, right? Okay, just a second, something happened. Is my screen moving? No, right? Can you all see my screen? Yeah, I think now it can. Sorry about the lag. Right. So what the Daniel adaptation model said was on this plasma membrane, you have protein here and here. But the final layer or the final model that we accept now is the Singer and Nicholson fluid mosaic model. And that's the accepted model, and that's the model that we study about. So now if you actually have to define the model, you follow what Nicholson and Singer said. It's a fluid mosaic model, which means that there is a dynamic nature of the membrane proteins and lipids. If you now remember what professor was speaking in her lecture. So your lipid or plasma membrane has lipid bilayer. Yes, there are two layers of phospholipids. You have the hydrophilic. Philic means loving. So hydrophilic head, which are moving, which points the outward side, and hydrophobic, which is inside, makes up the hydrophobic part of the plasma membrane. Okay, and then you have these proteins which are embedded in a mosaic. Okay, so you can see the proteins here that they are not just present on top or on the bottom. They are present within the like in that brick. So they are in the molecule, in the plasma membrane lipid bilayer, right? So they are not just over or below it. They are embedded like a mosaic, and they can move laterally. So the mosaic fluid comes from the idea that these lipids and proteins can move laterally, and that's what allows the dynamicity of your plasma membrane, right? And then along with this, what does the plasma membrane otherwise also consist of? You have lipids, of course, you have proteins. Proteins again are of two types, integral, which is intrinsic, and peripheral, which are extrinsic. Some are on the boundary, some are embedded in the bilayer. And then you have some carbohydrates, which are making part of the plasma membrane. So as you can see here, you have plasma membrane, you have carbohydrates, you have protein which is embedded, you have some proteins which are lying loosely, which are some, which, for example, these are peripheral, these are integral. Why they are integral? Because they sparse both the phospholipid bilayer. These are channel because they have this conduit from where they can allow molecules to pass through. These are peripheral, these are only at the edges. So they are only interacting with the molecules at the edges. These are globular. These are on the surface. They are interacting with other biomolecules. So what does it say? That the plasma membrane is very dynamic. It has several components in it and they all play important role, which together makes up the functional significance of the plasma membrane, like selective permeability. Plasma membrane is important for cell communication, for transport in terms of the diffusion, active transport, passive transport, and also cell recognition in immunity. Cell recognition is a phenomena which comes when you have important cell-to-cell attachment, you have fertilization process. All these are important and they take place via the identification. Thus, when this one cell identifies the other cell, and that identification happens through this plasma membrane. That's a very important part, and that's why it's widely researched and very well explained. Okay. Okay. So let's try to answer some questions. So this is basically the second last topic of the first part of the week one. So you have fluid mosaic model, who proposes, which I already mentioned right now. Then you have, I told you there are diffusion across the or movement across the plasma membrane, different kind of movement. So out of these movements, which of these do not require energy, and plasma membrane, if you have to mainly, if you have to define and tell, it's mainly composed of some biomolecule. Which of these would be the answer? And then which of the molecules enter TCA cycle? And I'll explain the TCA cycle's importance after we come to the answers. Singer Nicholson, yes. Diffusion. Okay. Lipid protein, pyruvate acetyl coenzyme A. Okay. Okay. Singer Nicholson, yes. Pyruvate, lipids means. Any more answers? Okay, acetyl coenzyme A. So I can see two type of answers for the last question. Okay, pyruvate and acetyl coenzyme A. Okay. Okay, so let me give you the answers. Right, so the first one everyone got right, Singer and Nicholson. Second one also people got right, lipid and protein. We've just explained that. Now here, uh, let me just check the answers first, what all people have written. So mostly they've written diffusion. Right, exactly. You've got that also right. Most of you, and actually every one of you, got the third one also right. The fourth one is where people are giving different answers, which is basically which molecules enter TCA cycle. The answer is acetyl coenzyme A, and I will explain to you why. To give you the joining links. But first, let me just tell you the other transports that are there. So, like the this question, which basically tells which movement does not require energy. Energy is when you have to utilize the ATP currency do to transport molecules across the plasma membrane. So active is basically the name. Active has to use energy in form of ATP to move substances. And this is because, why active? Because this is against the gradient. Anything which follows a gradient means anything which follows from high to low. So in natural conditions, or when there is transport take place is basically from high to low. So if you have a gradient in high to low situation, then it will be without the assistance of any or requirement of any energy, right? And active transports are basically those transports which is against the low to high, and for that you need a currency, right? Now, when you say endocytosis and exocytosis, which are basically again require energy, endocytosis means formation of vesicles and it basically takes inside the cell. So it is ATP required, and exocytosis is when you push it out of the cell. So it's again requiring ATP. So diffusion, on the contrary, they don't require any energy. There is very simplest passive process from high concentration to low concentration, right? And now coming to TCA, which touches upon the last topic of your first part of week one, which is about the introduction to cell basics, right? Touches upon cell energetics. And basically, most of you answered pyruvid, and people also answered acetyl coenzyme A, which was the correct answer. We will see why not pyruvate and where does pyruvate actually transfers or translates into the network. So we have mentioned all the different processes in the cells, right? You need transcription, you need translation, you need to move substances out of the cell, inside the cell, you need to replicate, you need to do protein synthesis, all this stuff. You require energy for them. So where do you derive the energy is what cell energetics talks about. So when you talk about cell energetics, you're basically trying to explain where is the cell deriving the energy that is required for all the different process that it does, right? So this energy is required for chemical reactions, for the transports and the mechanical work, and the currency here is ATP. Okay, so ATP is high, it stores very high energy phosphate bonds, and you hydrolyze those phosphate bonds and get the energy which is required for biosynthesis or active transport. Okay, so which are the, so any pathways that leads to the production of ATP is what you analyze when you're talking about cell bioenergetics. You want to understand how many ATPs are formed, how are those ATPs formed, how are those ATPs utilized. So it's all about the bioenergetics, right? So what are the different processes that are involved in cell bioenergetics? First is glycolysis. Glycolysis is when you have any food and the food is broken down. Your food is broken down to glucose, and then that glucose gets converted to pyruvate. Okay, that pyruvate, if you remember glycolysis, you will see that that whole chain of glucose getting converted to pyruvate is has its own 10 steps pathway, and it produces ATP, but not to a very great extent. You will remember it will produce only 2 ATPs. Okay, but you need more ATPs to function. So we'll see how does that function. Then you have the Krebs cycle, which is also called the TCA cycle, right? Now in this, people wrote that they are using pyruvate, but there's a step between your pyruvate and there's a step between your Krebs cycle, which basically converges pyruvate and makes it into an acetyl coenzyme A. And that acetyl coenzyme A is incorporated into Krebs cycle. And that Krebs cycle ultimately gives you more NADH and FADH2 molecules. Now, the confusion here was about the pyruvate. So just for your connection, in case of biochemistry, you will read it in more detail, where you will see that you have a glycolysis pathway and you have a TCA pathway, and then you have an ETC. All these together, the three of them together leads to the synthesis of ATP, or production of ATP. That's a step in between where your pyruvate is converted into acetyl coenzyme A, and that acetyl coenzyme A is taken into the Krebs cycle, and that is when your NADH and FADH is made. Okay. And since bioenergetics right now, I am talking about the processes where ATP is generated. I'm only talking about the first, third, and the fourth step. The second one does not lead to the synthesis of ATP. Okay. But when you come to your third step, which is your TCA cycle, where you do have ATP production, your first component that get incorporated is the acetyl coenzyme. Okay. Now, in the third process, which is your TCA, or also called the Krebs cycle, you have ATP, but you also have NADH and FADH production. These NADH, FADH also give rise to ATP when they are broken down. So that's how you meet the requirement of ATP in the last step, which is ETC. Okay, I remember in her explained this step in great detail. This was called the ETC, or electron transport chain, and she explained how the this was proposed by Mitchell, who told about the chemiosmotic synthesis, how you use the proton gradient to create the ATP. So I'm not going into detail, but I just wanted to give you an idea that this is what she was referring to. She was referring to the end step, but the steps preceding them are the glycolysis and Krebs cycle. And then you have the ETC, where all the NADH, all the FADH that was generated in the previous cycles are taken into the mitochondrial matrix, mitochondrial membrane, and there several complexes ultimately give rise to the, uh, so there is H2O, and then subsequently they produce the ATP. Okay, so uh, keep a note of that step and remember that. Okay, but all these together make up the cell bioenergetics. Okay, so as you can see on the slides, you have cellular respirations in animal cells, then you have the mitochondria, you have CO2 and water, right? And then you use the ATP. This also happens in plant cells who do it by photosynthesis in chloroplast. And they, instead of using the ATP, since they harvest the energy from sun, so in form of light energy. Okay, all right. So I have some questions which are pertaining to the bioenergetics part. So maybe I hope this explains this part a bit more. Okay, and then we move on to the next part, which is about DNA material, which is very interesting. Right. So this question says, which of the following statement best describes the role of oxygen in cellular metabolism? So in bioenergetics, what does oxygen act as? Anyone wants to take a guess? Option two. Anyone else? Option one, option two. Option two are lots. People are also writing option one. Okay, the answer here is it's a final acceptor in the electron transport chain where you will have the ultimate production of ATP. Hold on to that thought. I will show you the slide picture in the next slide. Before that, I have one more question. So remember your ETC. In ETC, you have several complexes, like I mentioned. So which one is the complex one? What does complex one do? Where does the ETC start from? Anyone else? Okay, just for people, just a reminder, in case you haven't attended NPTEL course before, so these lectures will also be available as YouTube recordings. The link will be already on the folder, I believe. So you will be given the folder which contains the PPTs, the PPT I'm using for this session. So the PPT will also be available to you, and the recording of today's session will also be available to you. So you will have both the sources available for your revisions when you are preparing for the exams. Okay. So I see a lot of yeah, that will be available on the app. You will be be given a link. So not on the app directly, but the should be included. Yes. Okay. Lot of tools on Swayam portal. Not directly. They will have the link, like I mentioned. They will in the Swayam portal also. They will give you a link to access the respective folders. Right. Lot of option ones and lots of options two. So let me give the answer here. The answer is option one, and that's because. So what does it say? It says what is the function of complex one. So if you define the function of complex one, it's basically to pump the proton into the intermembrane space and transfer the electrons to NADH from NADH to ubiquinone, which is Q. And this is your ETC. So basically it starts from this here, right? So you have your NADH. So like I said, the ATP synthesis is actually guided by chemiosmotic theory, where you have the proton motive force, which leads to the generation of ATP. And proton motive force means every time these molecules, NADH, FADH, they are uh, basically getting oxidized and they're releasing these hydrogen ions, the protons, the movement of the protons or the generation of the protons coincidentally coincides with the generation of ATP. So that's why these are energy molecules. So you have NADH, and the question said, if you go back here, the complex one basically pumps proton to the quinone so that that also can accept the electrons. So they basically need an electron acceptor that takes the hydrogen and electrons from them, and as a result, they have the edge. And then these protons later, you will remember the yeah, and ADHD hydrogens, right? So if you remember, professor would have told you names like F naught, F1, ATP mu, different which rotate and give rise to ATP. So that's a very well-defined structural process where you have the ATP generation because of this protons attachment. So you can see that. Okay, there are enzymes. There is an ATPase which gives rise to ATP synthesis. Okay. So ultimately, now the previous question, if you remember here, it said oxygen, what is the best role of oxygen in cellular metabolism? Oxygen comes in every step, but comes up also in the very last step where you have oxygen and water together making up hydrogen and oxygen together making up water and also leads to the generation of proton. And that's why this was the correct answer. It is a final electron acceptor in the ETC, which enables ATP production. And that's how this was correct. Okay. I did not go over this topic in much detail, but if you have any doubt, you can always put in the chat box. Otherwise, my suggestion is this is also very important topic in terms of cell bioenergetics. But with this perspective in this course, this might be just part of the first week. So we focus more on the cellular processes, which are basically replication and cell cycle. Okay. That's why I did not go over detail. Okay, now let's move to the second unit of the first week, and we have still 40 minutes remaining for the lecture, and I will finish it off very easily. Okay, so like we said, right, cells have all these macromolecules, DNA, RNA, protein, lipid. So the question was, and cells are arising from the pre-existing cell. So what is the genetic material that is getting transferred across the generation was always a question, and very well debated. We wanted to know if it is DNA or whether it is RNA or whether it is protein. And we have several beautiful experiments which are vividly described in our history and in the literature to understand how did they come to the final conclusion that DNA is a genetic material, right? So this is one of the experiment, which is called the Griffiths experiment, where we have something where we got to know of a phenomena, which is also very important for transfer of genetic material, right? And as you can see, so this is basically an experiment which was conducted on mice, and but with the use of a strain of bacteria. Which bacteria? This bacteria's name was Streptococcus. And what you see here is, just a second, yeah, Streptococcus pneumoniae. As you can see here, there were two different strains. You have R strain and you have a S strain. R strain is basically rough strain, as the name suggests. Even in endoplasmic reticulum, you have this R. So R is basically for rough. What does it mean? it is in comparison to smooth strain, which is the S strain. The S strain is capsulated, it has a capsule over it. And the rough strain, because it does not have this capsule, it's very serrated, it's non-capsulated, it's a rough surface. Okay, just the opposite of. And the S strain is virulent, which is basically causing infection, and R strain was a non-virulent form. Okay, now what did Griffith do? He injected into the mice different strains in different forms. So when a live S strain, which is basically capsulated strain, when it was injected into mice, as you can see, the mouse died. Okay, so something in the bacteria into the mice, and the mice died. When you have a live R strain, which is a rough strain, mouse lived. So basically, in this R form, it is non-virulent. The capsule is basically what is infecting. So because of the absence of capsule, R strain couldn't kill the mice. So mice lived. Okay. In the next one, as you can see here, he injected heat-killed S strain. So he basically heated the cells and then he injected these bacteria and the mice lived. Understandably so, that you killed the cells, you heated them, and something happened to the S strain and they could not kill the mice anymore, right? Now, the next one is the most interesting. What he did, he took heat-killed S strain, which is this strain, and the live strain, which is this experiment. He repeated, basically, he was doing these two together. He just took them together and injected. Now, this also led to living of mice. This also led to living of mice. But when you injected them together, the mice died. And what he also did, what he did was he isolated the blood from it, just the blood, and then he injected live strain with the blood in the mice also died. So something live strain is dying, but the moment you are putting heat kill strain, which is otherwise not killing the mice, when you put that with live strain, something is happening and the mice is dying. So what did Griffith say? Griffith said that there is some transforming principle going on, where from the dead S cells, it converted so the dead S cells converted the live R cells into a virulent S cell. Something is happening, and this phenomena was termed as bacterial transformation. What is transforming? The R strains, which are otherwise non-virulent, were transformed into a virulent form because of the S strain. Something was transferring. He did not know what. So what happened after that was, it was followed by this experiment, which was conducted by Avery, MacLeod, McCarthy. Okay. Now, what did these people do? They wanted to identify the chemical nature of this transforming uh, molecule, right? So what they did is they took the extracts from heat kill estrine and they treated it with protease, RNase, DNase with the idea that in each of these different extracts, if we say you will give RNA, so the RNA will be destroyed, everything else remains. And in the third one, we will give DNAs, it will destroy DNA and everything else remains. Okay, that is the whole idea about the experiment. So this is what they did, right? So you have the extracts from the S cells and you have done all of this treatment. Now you add R. Now, whichever replicates the situation here is what is important for the cell, right? Now, if you have added proteinase, you have no protein. You add R cells, the S cells appear. This is what you wanted. You need to convert these R cells into S cells, and they came up. So when you removed RNA, then also S cells came up. But when you remove DNA, this did not happen. So what it says that DNA is the most important for it to transform the R cells into R strains into the S strains. And that's how they concluded that DNA is a transforming material and DNA is a generating material. So the confirmation that we tell today that DNA is a genetic material comes from this experiment. Okay, so we know about this. Now, I hope this was clear to you. And we will just do quick 4 questions just to like strengthen the concept even more. Okay, a recap of what we just did in case anyone has any doubts. So the first question here is, why strain of Streptococcus pneumoniae is with the choice option. Show transform, which is easy question, which is a destroy the transform activity. So we already did about the Griffith experiments and the follow-up by Avery. So these are just questions which pertain to these experiments that we just discussed. I don't think it's easy. Are you able to see the slides? Yeah. Nice. Okay. I see the one different answer. Okay, there are some different answers. Okay. Okay. Okay, so I'll give the answers. Most of you have gotten it right. Okay. So once again, R strain, which is a rough strain, it is a non-virulent because of the absence of capsule, not DNA, because of the absence of capsule. It's non-virulent. First of all, it's non-virulent mean it is the native form, the way it is supposed to be. Okay, so it is rough. It has no capsule. S is S strain. The name S is because of the smoothness, and that's because of capsulation. Okay. Avery finally told the transforming principles of DNA, we all know, and that was confirmed because the enzyme destroyed the activity, and the enzyme was DNase. Okay, I hope people who have mentioned no DNA, if you have, then you just got confirmation of the answer. Yeah, but most of you have got that right. Every other question, right? So great job. Okay, so now quickly coming to DNA as the genetic material. So you have found DNA as a genetic material, but you also need to justify why DNA is genetic material, right? Why, how is it fulfilling the features of being a genetic material? So from several studies, it has been found that DNA requires several essential criteria which are required to store, express, transmit the genetic information, and also vary. Vary is very important because it also goes back to Darwin, who says that there needs to be variation so that you have a chance of surviving in differing environments, right? So you also need to vary. So that's also there, right? So DNA, if you have to name few of the features that are important for it to be a genetic material. First is its ability to store genetic information. As we know, DNA is composed of ATGC, which are nitrogenous bases, and these bases are the instructions for protein synthesis, for your cellular structure, for everything that happens downstream. They code these four nucleotides in different forms, encode the information for all the different proteins that we require for our basic survival, right? So it has the ability to store information. DNA is able to replicate accurately, and it also via the process of replication is responsible for inheritance of the genetic data. So it passes on the information to the next generation. Okay. Now DNA, one of the very important feature is it's a double-stranded structure. So it's very stable. RNA is single-stranded, so it has very high chance of getting degraded. You need a stable molecule that can sustain in the cellular environment. So you have DNA, which is more stable, and it's stable because of its chemical composition and structure. It has 2' deoxyribose in its structure, which makes it non-reactive, and that's how it is more stable. RNA doesn't have that. That's the structural difference between RNA and DNA, which makes DNA more stable. Okay, so you need to remember that. And like I said, DNA, which has all the information in a linear form, in form of its nucleotides, it is transcribed into RNA, which is translated into protein, and then ultimately the protein is what is responsible for all the other biogenetic pathways and all the other important cellular machineries, right? And where does the variation come from? These ATGCs can also undergo mutation to become another type of base, and that gives rise to mutation, and that gives rise to the variation that we see, right? And then it also has a very efficient repair mechanism. If you are a genetic material, you also need to sustain yourself so that you don't get frequently damaged and there is nothing left to perpetuate. So you also, DNA has a very good repair system as well. So it can uh, it can also have it, like it can do repairing in case it experiences radiation problem or chemical issues or replication error. Even its own machinery, when it is getting replicated, if there is error, you have machinery in the cell itself which can correct those aberrant. So DNA tries to keep itself very stable, right? Now, all this gives it a heads up as compared to RNA, which is less stable and single stranded, because of which you don't see RNA as a primary genetic material, right? So you need to remember these features, okay? And other things other than that you should remember are the four nucleotides and properties of the four nucleotides, of which one is purine, which one is pyrimidine, which are the nucleotides pair with each other, how many bonds are there, what is the direction of the strands of the two strands of DNA, the double helical structure. You need to remember the structure of the bases. So a nitrogenous base, you have phosphate, so you need to remember the sugar backbone. So all the structures you need to keep in mind. These are very important when you are doing the DNA structure part. Okay, right. Now another experiment which is of very importance when you are talking about DNA is the Michelson-Stahl experiment, which basically talks about the semi-conservative nature of DNA replication. Right? What does it mean? It means that basically DNA, like you can see here, DNA passes on its information into the next generation, right? So conservative would mean, so let's say this is parent one, parent, okay, like parent DNA, which is giving rise to the new DNA, and just see of them as a blue color strand. Now, one DNA molecule gives rise to two DNA molecules, okay? So a conservative mode will be the strands which were together in the parent generation stay together even in the next generation. So you have a new strand of molecule coming up in the next generation. So you have a red molecule coming up in the next generation. And then what will happen is in the next generation, with when this daughter becomes the parent, it passes on this one and they stay together and the other strand becomes red on its own. So this conservative means the copies which were together always remain together all throughout their generation replication. Now, the other ones as compared to this will be dispersive, which is basically you don't maintain the strand identity anymore. So if you have, let's say this one is one strand and you are transferring the information to the next generation, you have some parts blue, then some parts red, then some parts blue, then some parts red. So in one strand, you have both old and new information. Okay, so this is dispersive because there is no parental strand retention anymore. All the four strands which will come up in the next generation will have new and old information together, right? And semi-conservative is following the conservative mode, but uh, half of it, right? We'll see how. So you have two strands, and in the next generation, each of the daughters cells receive, or each of the DNA daughter DNA molecule receive one strand of the parent, and there is one strand from the new, right? So this is what is exactly the semi-conservative nature of DNA strand, and this is what happens when you see DNA replication. You have two strands in the parental generation. Each of these strands will be part of the new daughter cells. They won't go together. They each will be inherited in the next molecule. right? So that's semi-conservative. And this was proven by this experiment, which was done by Matthew Messelson and Franklin Stahl. Okay, they did this with E. coli, which is a fast-growing bacteria, as we all know. And what they did is, it's a beautifully designed experiment. So what they did is they grew the cells in heavy nitrogen, which is basically N15. Okay, so your DNA becomes very heavy. Okay, so they will first grow them in N15, and then they will later shift to light nitrogen medium N14. Okay, and to know how the DNA has been, or which DNA is part of their cells, they use the density gradients and ultracentrifugation, because that's exactly what will help them. Because the DNA have N15 and N14, which are differing in terms of their weight. So for them, using centrifugation method would have been useful, and they did that with a very sensitive method, which is CSCL, density gradient ultracentrifugation. Okay. So what this saw was that in generation zero, when they were heavy nitrogen N15, so there was only one band. Of course, because you have only one DNA molecule, right? So you have only one band, which was N15, N15. Makes sense. Now in the next generation, what they saw was a single intermediate hybrid band. Why intermediate? Because it had N14 and N15 together. So N15 and N14. So just for your ease, you can imagine 2 and 15 will have 15 plus 15, 30, and 2 and 14 will have and 14 and then 14, so 28. But intermediate is when you have 1 and 15 and 1 and 14, that will be 29. So they observed an intermediate phenotype, and they knew that something is not following conservative mode. But it became more clearer in the next generation, where they had two bands, one with N14, N14, and one with intermediate N15, N14. So they also knew this was not dispersive, right? So they were the one who, based on their the way their certification and the banding happened, they got to know that it was semi-conservative in nature. So this basically tells us that you inherit one strand of copy from the two strands. The two strands separate out. They are not together after one replication, right? So that comes handy when we are doing in the next session, when we will do DNA replication. All right. Okay. I think these would be towards the end. We are almost towards the end of the session. So let's just cover these topics with few more questions, and then just we'll end it soon. Okay. So Michelson-Stahl experiment, they prove DNA replication is what? The second is, they use which isotope of nitrogen? They use both, but you can answer which one they used first. Okay. And so when I say first, is basically which one did they use as a separate, as a variant, which is otherwise not in the natural condition. After two rounds of generations in N14 medium, the DNA observed was, so read this third question clearly and then answer. And the last one says, which method was used for them to separate the DNA? Mhm. Right. So most of you have written the answers. Anyone else? People are still typing. We'll just wait. Hybrid and 15 semi-conservatives. Yes. Okay. Um, okay. So these are the answers. As I can see, most of you got this right. Most of you got this also right, I believe. And 15 is what you all wrote. Yes. Most of you got this right. So this one hybrid and one night band, which you can see from this here. One hybrid and one night band here. Okay. Right. Cool. Okay. Now we're coming to the last topic, which is to be discussed for the first week. So now we know that you have DNA as a genetic material, and we know that they require to form protein at the end, right? So that together was formulated as central dogma, which said that you start with DNA, which replicates itself, which transcribes itself to gives an RNA, and which also then further gets translated into protein, and that forms the building blocks of life. But then you have an anomaly here. The RNA can also give rise to DNA, right? So this is happening via reverse transcription, where you have RNA which gets converted to DNA, and then this whole step takes place, right? And then which organism does this take place? This takes place in retroviruses. Why is this problem? Because we were telling that the material is DNA only, but in these retroviruses, you have RNA as the genetic material. So they have the central dogma there. So what explains them is the reverse transcription. These RNA also get converted to DNA, and then the whole procedure takes place, but they have their own machinery to do that. Okay. So as you can say here, this is the um, this is the machinery that the RNA uses to convert itself into a DNA, okay, via the use of a specific polymerase. We'll see polymerase, we'll come to these terms later again, but polymerase for reverse transcription is not the otherwise polymerase that is used for DNA replication. Polymerase, which is used for reverse transcription, is a RNA dependent DNA polymerase. So it's DNA polymerase because it makes DNA, but it is RNA dependent because it is using RNA as its template. Okay. And if we talk about reverse transcriptase, as you remember, professor also in her lecture note specifically dedicated some time to Professor Howard Remen, because he was the one who worked on this enzyme and he received the Nobel Prize for this. He was the one who suggested this entire reverse transcription process, right? And if you remember, he was basically working on retrovirus, which is also called the Rous sarcoma virus. And he was the one who found out that basically these viruses that basically use the RNA material. Otherwise, they are responsible for propagation by converting to a DNA form. Right, so this is very important, and he has done series of work that explains this method. Okay, so like I said, when he was working on his RSV, which is called the Rous sarcoma virus, what he saw was the viral transformation of the host. So when you say host cells, is basically, like I said, right, for them, after converting to DNA, for converting to protein, they have this own machinery of their own. Okay, and they need to have a host to continue their entire life cycle. Okay, so that's we'll see in this in a while. Okay, so basically, what Temin saw was there was viral transformation of the host cells. Viral transformation means the host DNA was becoming viral-like, and it was permanent. Means something, if something is transitionary or something is transitory, it will just stay in one generation. If it stays there for the entire generation and continues, also that's permanent change. So he saw transformation of the host cell. That's because, like I said, it's permanent because it was continued to the daughter cells. So when the host cell replicated, the daughter cells also had virus genetic information, right? And how is that possible? Why? Because host cells have DNA as their genetic material. These are viruses which have RNA as their genetic material. So how did the host DNA get an RNA component in that? Or what is it exactly? But this is a viral component for sure. Then what he did, he understood that these RNA were converting or were getting reverse transcribed into DNA, and that DNA was getting integrated into the host genome. You see how beautifully this entire mechanism has been evolved in nature. So you have these retroviruses, their RNA, they will first enter the host cell, become, they will, as you can see in this slide here, right? They will become, so the viral and reverse transcriptase, and it will integrate into DNA. Now, what happens when you get to host DNA? DNA will under replication as part of normal cycle. When it is getting replicated, the virus genome is also getting replicated. When the host genome is getting transcribed, the viral part of the genome is also getting transcribed. It's becoming a viral RNA. When it is getting translated, the viral RNA is also getting translated into viral protein, and the virus needs that protein for its infection. So when the protein has been made, the RNA will get the virus, out as a virus. So it comes into cells, gets integrated, and then is going to function. Now, why is because DNA is copy even in the new cells, so new cells will be having this DNA. So that's why these, because it is getting incorporated, a DNA molecule cannot have an RNA molecule. Something has to be a DNA of nature when it is getting integrated. And that's how he discovered that there is a reverse transcription which was taking place, and it was a very important and landmark discovery. As you can see, him getting Nobel Prize for this, right? So that was an anomaly to the central dogma, but it was ultimately shown that this is connected to the central dogma by its own process, right? So that's also very important. Right. And that will bring us to the last set of questions where we will, so now you will have some questions which were from the previous year's assignments. So do those, because you they will give you an idea of what kind of questions actually come in the exams. Okay. So yeah. So genetic material of retrovirus is reverse transcriptase synthesizes what? Integration of viral DNA into host genome is done by which enzyme? And I told you about retrovirus, but I did not tell you the name of the retrovirus. So can you guess which one of these is a retrovirus? Yeah. Mm. Anyone else? It's reverse transcriptase, okay? So the answer just now, just be clear of what you were typing. And that's the key, okay? Just look at the answer questions very carefully. Most of the times we know the answers, but we are just like, we don't read it clearly, and one letter here and then can make difference. Okay. When I say one letter, I really mean one letter, because I am talking about DNA, RNA. So if you're thinking about DNA and you're writing RNA, it matters. Okay, I will give the answers and then people can check if their answers was correct. Okay. So genetic material of retrovirus. Someone I saw wrote ssDNA. I hope you meant ssRNA, because retro means RNA. Okay, and it was single-stranded. Integration happens because of integrase. It's easy to understand. Reverse transcriptase synthesizes DNA from RNA. Okay, somebody wrote DNA from DNA. I hope you meant DNA from RNA, because reverse means that you are going in the opposite direction. So generally it is DNA to RNA. Reverse means it is the opposite way, RNA to DNA. Okay. An example is HIV, as most of you have already guessed. So great job. I hope people who I mentioned. I hope you just got that right, because that's what matters. Because like I mentioned in the beginning, it's a MCQ based question. So these are the basic mistakes that can happen, and then this might turn up in your ultimate results. Okay. All right. So now I have some set of questions, and we'll do it. We have 15 minutes, so we can finish it quickly. Okay. These are topics from the entire 4 lectures that we have in the first week. Okay. So we'll have different topics coming in different ways, come one after the other. So which are the following? This is all a revision for whatever we have done today. Okay. Which are the following statements our plasma membrane is correct? And since we have discussed it already, it should be very easy for you to answer all of these. Hmm. 3, 3, 3, 3. Yes. I see all of you remember already. Right. Fluid mosaic model, which is basically describing the membrane is a dynamic structure because the proteins that we remember, they are embedded in the phospholipid bilayer. Okay. As you can see, the questions are in different form. That kind of tells you that these are from 2 different years of assignments. Okay. So the modern cell theory does not include which of the following statement. So which theory, which of the statement is not part of the modern cell theory? Four, four. Okay. I see a general consensus on 4, which is correct. So modern cell theory tells that cells arise from previous cells or one or more cells. They carry hereditary information that is passed on to the next generation, and they are the fundamental and structural unit of living beings. Right? What it does not tell is that they form by free cell formation, which is not what it's right. Okay, next one. You see there is an A here and there is a B here. So you need to tell me which kind of bonds are these. Which option is correct, basically. Two. Everyone says 2. Oh, someone says 3. 3 also. Okay. I'll wait. Maybe I can just wait for a few more consensus. Anyone else wants to answer? I think 2 and I saw also 3. 3. What is 3? Hmm. Okay. Anyone else? All right. We'll just finish quickly. It's 2. I'll tell you why. So this is a water molecule. This is a water molecule. So this A is basically the bond between 2 water molecules. Okay. And in that molecule or in this bond, you can see that the bond is between a partially positive H and a partially negative O here, right? In this case, 2 water molecules, and they are connected by a partially positive molecule and a partially negative molecule. When you have interaction between a partial positive O and partial positive H and partial negative O, it's a classic hydrogen bond. So the first answer is hydrogen bond. And the second one, which is basically this bond here, sorry, it is again not moving, but the B one is basically a bond between O and H in the same water molecule. So when it is in the same water molecule, it is a polar covalent bond. Why? Because in this, O and H are sharing electron. Okay, and that is because of the electronegativity difference. And this is not ionic because they are sharing the electron. So this is a covalent bond, and the first one is a hydrogen bond. I hope. I also see some 4 and other options. I hope this makes sense and why this is correct option. Okay. If you have any doubt, please do type in the chat box. I will be happy to answer. Okay, next one. Activated carrier molecules store energy as. So where are their energies stored if I'm telling their activated carrier molecule? One. Okay. One. There's a consensus. Yes. Covalent bond. Okay. Like everyone has said, it's absolutely correct. It's energy-rich covalent bond. Just for explanation, if someone has any doubt. So when I say activated carrier molecule, I am talking about all the molecules that are carrying energy. For example, ATP, NADH, FADH, acetylcoenzyme A. So all these different molecules. Now, in all these different molecules, there is high energy covalent bond. If you see ATP, they have a bond between the phosphate and the anhydride group. If you have NADH, you can have covalent. You have covalent bonds. Again, acetylcoenzyme A. If you see the structure, they have covalent bond. So all these covalent bonds, when they are broken, they have basically the high energy. So they have the energy generation, right? Why the other options are incorrect? So if you see what it says that it is rich in hydrogen bond is a weak bond. It says energy rich iodic bonds. Ionic are basically the bonds which dissociate in water. So it's not what it meant like here, which is activated carrier molecules. And hydrophobic bonds are not energy carriers, they are entropic, right? So that's why it's covalent bond. Okay, next one. Which are the following modes of mobility of membrane phospholipid? Remember, I told you that it's fluid, but which is the mode of the mobility of the membrane phospholipids and which is the most energy consuming? And you can think why is it which one, even if you don't like know the answer, you can take a guess just by the mode of the movement of the proteins, which will require more energy. Yes, people are very smart here. Yes, flip flop, right? Just imagine. So this is basically when I'm talking about phospholipid movement. If anything is laterally moving, it is very fast. It just needs to move like this. So it's very fast. It doesn't require much of the energy, right? If you're rotating, you still have a central axis. You just rotate, you have very low energy. Flexion is basically bending of fatty acid tail. So in phospholipid, you have the fatty acid tail, it just bends. So it's also very fast. But in flip flop, the name suggests that is basically you are moving from one direction to other. So one leaflet is moving from one side to the other side. This is very energy consuming because you are basically changing your direction. So every one of you got that right. Very correct. Okay, next one. Pick the correct option. So read the options and take a guess which one is the correct answer. Anyone? One? Okay, anyone else? Okay, see 1 and 3. I will give you the answer. That is 1. So basically, as ion transporters actively move ions against the concentration gradient. It's correct because it is moving ions. Whenever it is against the concentration ions, it is actively moving it. That's why it is correct. The second statement says it does not allow the ions to diffuse down the concentration. That's not correct. Ion channel are basically transporting ions. Okay, they can do it down the concentration gradient or against the concentration gradient both. When they're doing against, they are actively doing it actively by using ATP. Ion channel creates ion concentration? No. Ion concentration gradient is because of the ion concentration on the both sides, right? Ion channel dissipates this because it allows the movement of ion. Just this is what the distinction is. And ion transporters cause selectively permeability to ions. That's not correct. It's the ion channels. Transporters are the proteins in those channels that basically take the protein. The channels are what makes it the selective permeability. Okay. And you almost everyone got right. So that's great. Okay, now we come to the second part, which is DNA. Again, quickly, topics which I couldn't cover. In the Watson-Crick model of DNA, the two strands of double helix are held together by which type of bond? Easy. Remember A to D and G to C? Yes. Which bonds are holding these nucleotides? Yes, as you all go. Hydrogen bond between complementary nitrogen bases. The central dogma of molecular biology states that genetic information flows from DNA to RNA. Which of the following step is responsible for transferring information from DNA to RNA? In the central dogma, which step refers to this part? So there is replication, there is something else, transcription, translation. So which one is correct? DNA to DNA, DNA to RNA, RNA to protein. That's the central dogma. It's DNA to RNA, okay? Remember that. Okay, I will give the answer. This is transcription. So replication, DNA, but basically just taking the information down. So you're just transcribing. You just whatever information was there, you're transcribing the information. Why is it transcribing? Because if DNA is ATGC, RNA also retains ATGC. The only information difference is that T becomes a U in RNA, except that everything else. If the original sequence was AATTGGCC, the RNA will also have it in the same sequence, AAUUGG. CC. Okay. So it's just transcribing the data. So it's transcribing, right? That's why it is transcription. The third one is translation when your RNA becomes protein, because there AAT, ATA, and TTA will be different information, and it is not exactly identical because if you have stop codon, the protein sorts there only. So it is translating the information. You are basically, what is translation? If you have, let's say, you're talking in another language and you want to translate the language to another third per language, you will basically change the words and use it accordingly in your language, right? So you're translating the word, you're translating the language. So RNA has AUGGG, nitrogenous base, but protein has amino acid. Your three nucleotides together code for an amino acid. You're translating the job, you're translating the language. So it's translation. You can remember it like that. That would be easy for you to remember. So it's translation. Okay. And the fourth one is mutation. It's basically a change in the nitrogenous base, in a change in the sequence, nitrogenous base or protein or anywhere. Right. So that's mutation. That is not what it's referring to. The question was DNA to RNA. So that is transcription. Okay. Okay. Next one is again very easy. This is the second time this question is coming. In the Avery MacLeod-McCarty experiment, which macromolecule was called the transforming principle? Very easy, right? We've already done this multiple times now. Okay, we have only 1 minute remaining. I will just quickly do the remaining question. Won't take much time. DNA. Okay. Methyl salt we have already done. It's semi-conservative. Okay, this one. What is the role of messenger RNA in protein synthesis, which is basically translation? Anyone? Because this video don't complete. Would studying the video lectures and notes be sufficient for the exam? Yes, yes, yes. Your syllabus will be part of it, but that's why you have this problem solving session where we try to bring you question from the previous years so that you get an idea what kind of questions will come from those syllabus. So do come up to this video problem solving session. If you can't attend it live, please do look into the YouTube videos because we'll give you these questions from previous years, which will give you a understanding how to go ahead. Right. Okay. A three, right? Yes. I'm sorry. Yes, three. Because mRNA is what is transcribed from DNA, and that is giving you a template to code for the protein. Okay, we'll do replication anyway. So when there we'll understand it better that the enzyme which is responsible for opening up is helicase. Okay. Yeah, these are the questions I was telling. I will just quickly go over the questions because time is up. I don't want to hold you up. So it says the bond between sugar group and nitrogenous base is what? Okay. So these are different kind of bonds that are important for DNA molecules. So you should remember them. Give me 5 more minutes and I will wrap up this session. I just want to show you the question so that when you are looking at the answer, you know which answer is correct. Okay. Yes. You guys are saying phosphodiester bond, but that's not correct. Phosphodiester bond is between two nitrogenous base, like in a chain between two subsequent nitrogenous base. Glycosidic bond is between sugar and nitrogenous base. Okay, that's why I wanted to tell this. And this is from your section where Professor discussed about reverse transcriptase. So do you remember what does actinomycin D do? Yes. This was for the discovery of the reverse transcriptase. Okay. So then when he used, sorry, actinomycin D, which inhibits DNA-directed RNA synthesis, he could see there was no transcription anymore. Okay. And the viral protein was not able to come out of the cells. Okay. Right. This is what I wanted to come to. That's why I was holding you up. So when you are doing DNA as genetic material, remember about these rules, Chargaff's rules, and there are certain set of rules that are there. Out of these options, you can you guess take a which one, right? So I hope this gives you an idea what kind of questions come. Now remember, MCQs in the final exam, definitely it will be over the syllabus, like all parts of it, but having these question banks help you. Yes, three, as most of you are guessing. Next is, what is the difference between deoxyribose and oxyribose sugar? The basic difference why it makes DNA and RNA structurally different and also contributes to the stability of the biomolecule. One, right? Most of you are writing one. That's correct. Right. So deoxyribose sugar contains hydrogen at the C2 position. Ribose sugar contains hydroxyl, and the name suggests, right? Deoxy means without oxygen. So at C2 prime and prime position, you have no oxygen in DNA, which makes it less reactive, which makes it more stable. Okay. Yes. So what is the direction of the phosphor? Some people wrote phosphodiester bonds, right? So now that you know what is phosphodiester bond, do you know the orientation of the phosphodiester bond between two nucleotides? Yes, two. See, one and two both. So anyone, I will take more consensus on this. Okay, I will give you the answer. Most of you have written one, a few people have written two, and I understand why. The answer is C3 of first pentose sugar to the C5 of the second pentose sugar. And I know people say that we have 5 prime to 3 prime directionality. When you look at the structure, this still holds up, but the bond is formed between the C3 of the first sugar to the C5 of the next sugar. Okay. All right, and the last question about this is also this is very easy actually. This is the base pairing that happens in DNA. Extremely sorry. Just 1 second. I'll just share it again. Yes. Yeah. Coming back to it, you all got it right. 4, right? Okay. So that brings anyway to the end of the session. Okay. Um, so this will also be there in your uploaded videos, and I have a few more slides which actually explains bonds that we are just talking about. So you can find them in the PPT. All right. Okay, that brings an end to this session, and I'm sorry to hold you up for so long. If you have any suggestion, do let me know. Otherwise, we meet each other in the next week, next Monday. Thank you so much for being part of the session. Thank you.